Recording. Go ahead, Jesse.
Okay, great. Well, welcome everybody. It's my absolute pleasure to welcome Professor Katia Koelle, who is at Emory University, joining us from Atlanta via Zoom. So really pleased to have her today. I know that some of the students, or all of the students, will have, or sorry, all of the first year students. Some of the students will have read one of her papers already. We're excited to hear more. So Katia has done really great work, not just on SARS-CoV-2 evolution and phylodynamics, but really on a bunch of other viruses. So developing some very interesting theory and models and evolutionary dynamics. And she did her bachelor's at Stanford, followed by a PhD at the University of Michigan, Ann Arbor, before starting her faculty position at Emory, where she is now. So thrilled to have her. And Katia, I'll let you take it away.
Great. Well, thanks, Jesse, for the introduction. Can everyone hear me? Great. And can people see my screen?
Looks good.
All right. Great. Let me move all the the Zoom parts here so I can actually, can everyone still see me in my screen?
Yep.
Yes.
Okay, fantastic. Okay. So what I'm going to be talking about today is some work over the last year and a half from my group, which my group has, over the last 10, 15 years has focused almost exclusively on flu and, a little bit of dengue, a little bit of norovirus. But about a year and a half ago, somewhat unsurprisingly, we shifted to looking at SARS-CoV-2. And so we have various projects ongoing, looking at how SARS-CoV-2 evolves and how that evolution has implications for understanding viral spread at various levels. So I actually love to have a little bit more interaction during my talks. So there's a couple of places where I'm going to actually ask questions, and hopefully you guys will not. Hopefully you guys will actually, respond and be interactive. But there's just a couple of slides like that. So I'm giving you fair warning.
I can also interrupt you. Do you mind being interrupted?
Oh, please, that's great.
Okay. So there might be a few questions that will come in the chat or whatever. And yeah, we'll keep an eye on them.
Yeah, one thing with these Zoom seminar talks, it's really unfortunate because the speaker usually can't see that many people, right? Usually it's only a couple of people. And then oftentimes people also turn off their videos, of course, for bandwidth reasons. But it's, I generally really love giving seminar talks in front of real people. So anything to kind of make it more like that situation would be fantastic. Okay. All right, so I want to start off just with talking a little bit about viral dynamics across scales. And so my group studies kind of viral, effectively population dynamics across a wide range of scales. So we're interested, for example, in trying to understand the relationship between kind of viral input into a cell, right, and how much actually kind of comes out of a cell. So kind of viral dynamics at the level of a specific cell. Also kind of spread within an individual host. At transmission events within populations and across multiple populations. So we're really interested in those dynamics. And if you think about viral evolution or pathogen evolution, it can also be characterized at these distinct scales of organization. And what I'd like to talk today about is actually some work really spearheaded by two of my graduate students who are both phenomenal, Mike Martin and Youngsun Park. And I'd like to talk about two different scales here of SARS-CoV-2 evolution. The first occurring here at the level of kind of the transmission event, and one kind of occurring here at the population level. So starting off at kind of this transmission event scale, one thing that we've been very interested in my group has been on looking at transmission bottlenecks, right? And so what are transmission bottlenecks? So we can imagine, during a transmission event, we have an individual who's a donor, right, who's infected with a viral pathogen, and they're transmitting the viral infection to a recipient, right? And so, if we think about kind of a donor individual who's infected with, the virus population, can be genetically somewhat heterogeneous, right? Which you can see here depicted as these different colored balls, right? And then during the transmission event, there can be what's known as a transmission bottleneck. So you can see this bottle here. So a few different viral particles might, come into this cup, right? Some of those particles will kind of die, some will survive, and some will kind of seed the next generation kind of in that recipient, right? And so what we've been interested in has been both developing kind of statistical methods as well as kind of applying those statistical methods to virus sequence data, trying to estimate what this transmission bottleneck size, or what we call NB, is. So it's just to define it, the transmission bottleneck size is the number of virions or viral particles that go on to really establish a genetic lineage in an infected individual. So what it's not the, if someone's coughing, someone who's infected is coughing onto someone else, it's not the number of virions that kind of fall on kind of that recipient, right? It's also not the number of unique virus genotypes that establish infection, but it's the number of viral particles or virions that go on to really establish a genetic lineage in an infected individual. So why are we actually interested in quantifying this transmission bottleneck? We're interested in this for various different reasons. One is because it has implications for understanding within host dynamics and disease development. It has implications for identifying who infected whom in a transmission cluster. And then it also has implications for understanding rates of population level adaptation. So what I'm going to do in the next few slides is actually go through each one of these implications and how transmission bottlenecks actually might be relevant in each one of these three cases. Okay, so first talking about kind of within host dynamics and disease development, this is going to take a couple of slides. So there's been within kind of my area of research, there's been kind of a longstanding interest in understanding inoculum dose and how that relates to within host disease dynamics and symptom development. And so this is an example out of Yoshikawa's group at Wisconsin, where they took these, for SARS-CoV-2, these Syrian hamsters depicted here, and they exposed them to either high dose or low dose of SARS-CoV-2. So in either case, these doses are pretty high. But the high dose individuals, you can see that those hamsters actually lost more weight before recovering than the low dose ones, right? And if you look at the severity of their infection as quantified by basically lung scans and damage to the lung, individuals who got a higher dose of SARS-CoV-2 had higher levels of disease severity before recovering than low dose individuals, as you can see here in blue. Right, so the idea here is that inoculum dose, the higher the inoculum dose, the more severe the symptoms might be. Okay, so this has led to an idea that some of you might have come across in the popular media, which is on the effect of masking actually on potentially disease development or symptom development. So this has been termed the variolation hypothesis, primarily by Monica Gandhi at UCSF. And so the idea here is that if individuals wear masks, right? So masks have been demonstrated to decrease the probability that if you're infectious and you're wearing the mask, it decreases the probability that you're going to get someone else infected, right? And it's also going to protect you from getting infected yourself if you're uninfected, right? But the idea here is actually somewhat different. The idea here is that if you're wearing a mask, if you do happen to get infected from someone else who's infected, that the number of viral particles that are actually infecting you might be fewer than if you were not wearing that mask, right? And so with a fewer number of viral particles, that your disease severity might actually be lower because effectively you're getting a lower inoculum dose, right? So kind of like in this case of these Syrian hamsters. Right. So this is what's known as the variolation hypothesis. And you can see it kind of as these kind of opinion pieces. The idea is that we can wear face masks to protect us not only from infection, but if we do get infected, it will protect us from disease, from severe disease. And it's called the variolation hypothesis because it dates back to kind of pre-1700s with smallpox. Smallpox killed about 1 in 3 individuals who actually got infected, so a 33% mortality rate. So the idea with smallpox was that people who got infected with smallpox and actually recovered from it, sometimes they would have these kind of scabs on their skin, and those scabs could be kind of picked off, pulverized to a powder, and then as you can see here, that powder can be, for example, like shot up someone's nose, right? And that person would develop an immune response to that virus, but they would not actually have as high of a chance of dying. So I think the chance of dying here is something like 1% rather than, or 1 to 5% rather than 33% chance, right? Which is much better. So that's known as the process of variolation, and this was adopted, started in Asia, and then was kind of brought over into Africa and into Europe. And the idea here was variolation worked because it induced an immune response, but the number of active kind of viral particles that someone was actually exposed to was fewer, right? And so they wouldn't get as severe disease, right? But still have an immune response. So again, brought over into SARS-CoV-2, maybe wearing masks actually reduces the number of viral particles that you're actually exposed to, and so lowers inoculum dose and then lowers your probability of developing severe disease. So if we think about the transmission bottleneck, right? So if the transmission bottleneck is extremely large, right, you can imagine that wearing masks might reduce the number of viral particles that get across, right? Lowering inoculum dose, so you get less severe disease, right? However, if the transmission bottleneck size is already very small, that's much less likely to be the case, right? Because if it's 1 or 10 viral particles, then it shouldn't really matter whether you get exposed to 10 viral particles, for the transmission bottleneck, or it's 2, right? If you're wearing a mask, that's still a very small number, right? Okay, so the second reason why we care about transmission bottlenecks is because they are useful potentially, because viral genetics can potentially be used to actually identify who infected whom in a transmission cluster. So if you think about kind of spread within a transmission cluster, like let's say in a healthcare setting, it might be really desirable to know whether 10 individuals who got SARS-CoV-2 within a hospital setting, whether it's likely that they all got it from the same nurse or whether they transmitted it between one another, right? So that we can inform public health strategies, so maybe more protection kind of for the nurses who are interacting with the patients, for example, versus if the transmission is happening between patients. So there was this idea, especially early on, the idea being that we could potentially take samples from infected individuals, and based on those samples, we could actually reconstruct who infected whom. Right. So now if we think about transmission bottlenecks, we could think of like, well, when would that actually be possible, right? And so you can imagine if the transmission bottleneck sizes were really large, right, that might be a possibility because you have a lot of genetic variation, right, present in a single individual, and if a lot of it gets transmitted, right, then you can see, oh, this person got it from this person, right, or if only a subset of it gets transmitted. However, if the transmission bottleneck size is very small, right, then what's going to happen is within a transmission cluster, they're all going to look pretty similar, and that they're all going to have these variants, which are kind of unique to themselves, right? But that it's going to be much harder to actually reconstruct it who infected whom. Okay, and the third reason we're interested in the transmission bottleneck size is because it really impacts rates of population level adaptation. So you can imagine here, so here we have the donor who has kind of a lot of this genetic variation, as you can see depicted here. If the transmission bottleneck size is loose, a lot of that genetic variation gets across, right? But if it's tight, a lot of that genetic variation gets lost. So you can imagine in an infection, let's say a beneficial mutation arises in a donor, reaches maybe 5%, 10%. If the transmission bottleneck size is tight, right, then there's a good chance it actually gets, that beneficial mutant doesn't actually get transferred, right, which slows down the process of adaptive evolution for the virus. And so transmission bottlenecks, very tight ones, are really kind of good news for us effectively, because it slows down population level adaptation of the virus. Okay, so now that you might actually care about what the transmission bottleneck size is for SARS-CoV-2, I want to talk a little bit about how it's actually estimated. So there are a variety of different methods available, or that exist in the literature. We developed kind of one ourselves a few years ago. And the idea here is that we have, in a donor individual, we have a sample from that donor individual. And what we can do is we can deep sequence that sample, right? And so you can see these reads. We can kind of map to a reference strain. And then what we can do is go along the genome, and for any site that has, that's polymorphic, so has more than one allele, we can basically determine the proportion of the reads that have kind of one variant versus the other variant. So, what proportion have A versus G, right? And we can do that for every polymorphic site in the genome. If we think about, if we have information on the donor recipient pair, right, then we know that there's been a transmission at some point. We have the founding viral population size, right? And then we have kind of growth of that virus population until we can kind of sample that population. And we do the same thing in the recipient. So the idea here is based off of these variant frequencies, right? In the donor as well as in the recipient, what we're interested in is estimating this over here, which is the founding population size or the transmission bottleneck size. Okay, and the method that we developed, I'll just point it out here. It allows for this growth process to be kind of a stochastic growth process, and it's effectively a stochastic birth-death process. And statistically, this method allows for, accommodates variants that fall below the variant calling threshold. So this is the variant calling threshold. So basically what you have in these deep sequencing data, you have sometimes kind of a bunch of errors, right? And so generally what's done is you kind of, you set a threshold of 1% or 2% or whatever percent, right? And you say anything below that, you don't know whether it's real or not, right? It could be real, but it could also be sequencing error. Okay, so this is where your input is needed. Okay, so just an example to provide intuition. So let's say we have over here in the donor, we have these two sites that have where there's genetic variation present, right? And then in the recipient, we at those same sites, we look at the variant frequencies at those sites. So if there's a large bottleneck, so let's say we have two variants in the donor, one at this frequency of maybe, I don't know, 40%, and one at, let's say, 10% or so, right? If the bottleneck size is large, what frequencies do we expect those variants to be at in the recipient?
Don't be shy.
There's an answer in the chat saying, okay, similar to the donor, same frequency.
Yeah.
On 80-20. Okay,
so so that first answer, which is similar to that of the recipient, that makes a lot of sense. So if there's a large bottleneck, right? Let's say a variant such as, so let's say we're looking at this site over here. If you look in the upper right, so this blue, or let's say this purple variant is present at 50% of the population, the viral population, the donor. If the bottleneck size is very large, right, then you would expect about 50% of the reads kind of in the recipient to be that purple variant, right? And if it's 20%, in the donor, then we expect it's about 20% in the recipient, right? And so what we expect if the bottleneck size is very large is that the donor variant frequencies or the recipient variant frequencies are very similar to the donor variant frequencies, right? And so what we expect is actually kind of this dot, these points to kind of fall on this x equals y, line, right? But if we have a small bottleneck size, what do we expect then? Should open a chat again. Step function. What do you mean by step function, Lucas?
Small frequencies get floored to 0 in the recipient, but after a certain threshold, they get floored to like 1.
Yeah. Yeah, absolutely. And so let's say there's something, let's say that transmission bottleneck size is 1, right? So very small. Then if you're at a 10% frequency here in the donor, right? Chances are, you know, that the single viral particle that kind of makes it across to establish, you know, in the recipient, chances are that it's probably not going to be that 10% variant. It's going to be the 90% kind of wild type one. And so what you expect is to have kind of a point down here, right, at 0.1, 0, right? And so you expect to kind of have a smattering of points down here, maybe kind of 1 up here, but effectively kind of either at 0 or at 1. Right, in the recipient, and the higher the frequency in the donor, the more likely it is to be kind of at 1 in the recipient. Right, great, thanks. Okay, so let me go on. Okay, so now that we've kind of gone through that example, what I want to do is kind of present our analysis or our reanalysis of some analysis that was done looking, which had the goal of estimating the transmission bottleneck size. So what we did was we looked at this paper when it came out. This was one of the earliest papers that came out that tried to estimate the transmission bottleneck size. And what they had was deep sequencing data from over 500 virus samples. They had a lot of surveillance going on in Austria, and so they were able to identify transmission pairs, who infected whom. And what they did in this paper was they estimated the transmission bottleneck size to be about 1,000 virions. And when we read this, we were kind of surprised by it to begin with, because there was some really nice work out of Adam Loring's group a few years ago that quantified, using that beta binomial approach that I just mentioned, that the bottleneck size for seasonal flu was on the order of 1 or 2 virions, so very, very few viral particles. Right? So for SARS-CoV-2, that it's that different was really surprising to us. So that's why we decided to kind of take a look again. These are their results from this Science Translational Medicine paper. So they have 39 established transmission pairs. And what they did was they looked at various or a couple of different variant calling thresholds, right? So they say, okay, well, anything that's above 1%, we're going to call real, right? So at a 1% variant calling threshold, what they found was on this x-axis, it's the transmission pair, and you can see, you know, from here, for example, 003 to 143, 003 to 145 or 146. So that's the donor giving it to that recipient, and sometimes it's the same donor giving it to multiple recipients. And this is their estimate of bottleneck size using that beta binomial method I mentioned. And what you can see here is that there's a huge amount of variation, right? So there's some transmission pairs that have, you know, over 1,000, you know, more on the order of about 5,000 viral particles estimated, and then there's some that have lower. You know, so we see a huge amount of a huge amount of variation between transmission pairs. But what really caught our eye was that if you go up to 3% variant calling threshold, which they did, and this is a supplemental graph that they have, what you see is sometimes this really precipitous drop from kind of larger transmission bottleneck sizes to smaller ones for just certain, and you can see that here very well as well. And you see this happening for some transmission pairs, but not for other transmission pairs. But there was this really precipitous drop, which we were really concerned about. So what we decided to do was we plotted kind of their results as a function of maximum donor allele frequency. So what does that mean? So here we have a donor, here we have a recipient on exactly kind of the plot that we just looked at before. This site is a site that has the highest variant frequency in the donor, right? So basically there's a site where about 20% of the reads have, let's say, a G, and 80% of the reads have an A, let's say, right? So on this graph, this is the maximum donor allele frequency, If we plot all 39 transmission pairs bottleneck sizes against the maximum donor allele frequency, right? What you see is at the 1% cutoff, we see these black estimates over here. If we plot their 3% cutoff estimates, what you can see is for each one of these transmission pairs that actually had, high maximum donor allele frequency, we saw this kind of precipitous drop in the bottleneck size estimate, right? And another pattern that you can notice is that all the ones that have very low maximum donor allele frequencies have very large transmission bottleneck size estimates, right? So this indicated to us two things. Well, one thing really is that the really kind of low frequency variants, the ones that were below 5 or 6%, are potentially telling us a very different story from the higher frequency variants, right? And we can see this here going from 1% to 3%, because if you go from 1 to 3%, if you're lopping off kind of the variants that are between 1 and 3%, right? And you're getting major reductions in the bottleneck size, right? That's indicating that those low frequency variants are telling you that the bottleneck size is very, very large, and that the higher frequency variants are telling you that the bottleneck size might actually be quite small. Okay, so this is concerning because if there's kind of a true bottleneck size, right? The variants should, regardless of what allele frequencies there are, should be telling you the same thing. Okay, so what we started to look at then was these individual plots like this, where we have donor on this axis, recipient on this axis, and what we found first is by looking at these low frequency donor variants, oftentimes they would be on this kind of 1-to-1 line, right? So this is just an inset of this larger graph. And so variants that are really between 1% and about 4%, or even 5%, are telling us that there's an extremely large bottleneck size, because if they're observed 2%, in the donor, they're pretty much observed at 2% in the recipient. They fall on this 1-to-1 line. However, as you can kind of see from this plot, the high frequency variants over here are much more in line as what Luke said before, kind of the step function, which indicates that there might be a very small transmission bottleneck size. Okay, so these variants are telling us different things, right? And now I want you to look at this point over here. This point over here is a variant which is absent from a donor. You don't see it in the donor, but if you look at the recipient, it's fixed in the recipient. So all the reads have a different nucleotide at that site than from the donor. And this can come about if during the transmission process there's kind of a small number of viral particles getting across, there being a mutation that occurs very early on during the replication of the virus, and then that new viral variant actually kind of fixing in the population. Okay, but you can imagine if that's kind of fixed in the viral population, it must have occurred in some genetic background, right? And then all the other so it indicates that if it occurred in one genetic background, right? And so if you don't have rampant recombination going on, then there should also not be any shared kind of subclonal genetic variation even present between donors and recipient, right? Because if you have kind of that one variant fixing, it should have brought along kind of all the other, whatever genetic background was in, it should have kind of brought to fixation all that, all those kind of alleles that it kind of was in the genetic background of, right? So this really indicated to us that these, that these variants here at very low frequencies were probably spurious, right? It's really this point here and this point that were telling you very different things from these points over here. Okay, so what Mike did at this point was we just looked across all the different samples that we had for the study. And he looked at the sites that were polymorphic and these variants that were present. And what he found was that from the most abundantly, or the variant that was most commonly observed across all these samples, it was found, it was this variant here, which was this G 25,135 T variant, it was found in all 43 samples. And if you looked at the frequency it was found in, it was found between kind of 1% and 2% frequency in all of the samples that were considered. Right, the next most common one was also found in 43 samples, and again at kind of between 1% and 2%. And so, and some of these samples were from Western Austria, some were from Eastern Austria. So it's very unlikely that that this was actually kind of shared genetic variation. But this actually indicates that very likely these were, these are spurious variants. And that they occur at very, very similar frequencies. And so it might be part of the, some kind of sequencing protocol kind of error that's kind of come back. So what we did is we found kind of these up to about 6% or so. So what we did was we re-estimated using that beta binomial approach, we re-estimated the transmission bottleneck sizes across these 39 transmission pairs. And if we go up to a 6% cutoff, right, what that should do is it should not really kind of change the maximum likelihood estimate, but it should actually just increase the confidence intervals, right? Because you just have less data to work with. But what you see here is if we again look at maximum donor allele frequency on the x-axis here with a 6% cutoff, what happens here is that our estimates actually go down to 1 to 2 viral particles, right? Which indicates, again, kind of if we are removing the 3 to 6%, that the 3 to 6% were kind of inflating our transmission bottleneck size estimates, and those 3 to 6% are likely these spurious variants. So we think actually using the 6% cutoff, right, even though it increases our confidence interval range, is much more appropriate for actually determining transmission bottleneck sizes, and that anything below 6%, at least in this study, could likely have included these spurious variants that actually artificially inflated the bottleneck size. So what we then did was we tried to look across the different transmission pairs that we could still look at, and come up with kind of an overall estimate of transmission bottleneck sizes. And so we did that by combining data from these different transmission pairs and using kind of a zero truncated Poisson distribution and estimating the parameter from that. And what we found is that our estimate is that about 80% or so of transmission events involved only a single viral particle being successfully transmitted to a recipient. About 18% or so involved 2, and a much smaller number 3 or more, right? And so about 99% or so of successful transmission resulted in 1 to 3 viral particles being transmitted. So we can go back to kind of our implication slide, which is, if we only have 1 to 3 viral particles being transmitted, very similarly to seasonal influenza, what is the plausibility of this variolation hypothesis? The data that we looked at was from Austria before people were wearing masks. It was very, very early on during the pandemic. So we don't think actually that if in an unmasked setting, if 1 to 3 viral particles are actually getting across to found an infection, with masking, this makes sense in terms of masking actually reducing the probability of transmission, right? But we don't think that wearing masks would actually have really an effect on disease severity, because if an individual gets 3 viral particles versus 1 viral particle to start off the infection, it's not going to make a big difference, right? So we don't think that masking will actually really impact disease severity, contingent on infection. Second thing, with our viral genetic data useful for identifying who infected whom, we think it would be very difficult, if not impossible, to actually use virus sequence data within a small transmission cluster to figure out who infected whom with such low transmission bottleneck sizes, with such tight transmission bottleneck sizes. And lastly, we think there's kind of a great kind of constraint on population level viral adaptation with such a small transmission bottleneck size. Okay, which leads us to, okay, we know that all these viral variants are circulating. We've got alpha, we've got, delta, et cetera. And so how is virus actually adapting at the population level if there's such small transmission bottlenecks? So the way kind of I think about it was really, I think, really facilitated by conversations with Adam Loring, who, when he had given a talk at Emory a couple of years ago, talked about it in the context of flu, and thinking about it in terms of these, in terms of jackpot events, I guess Las Vegas, like kind of events where you have, each individual could be considered a slot machine, right? And mutations can arise kind of stochastically within an individual, and they might actually fix kind of like you have here through genetic drift, right? So they can fix by genetic drift, but individuals that carry these mutations might have higher transmission potential. So we can imagine that, if we have all these independent slot machines, there's just so many slot machines that are being played that there's these beneficial mutations or kind of these jackpot events that kind of occur very rarely in some individuals or in some slot machines, right? And then those slot machines, right, effectively can kind of infect more slot machines. But that mutations don't arise within a single individual, through positive selection, right? But it's really this crucial role for kind of genetic drift in terms of fixing at the individual level beneficial mutations. Another possibility for SARS-CoV-2 that's been mentioned is the role that chronic infections in immunocompromised individuals might play in terms of chronic individuals getting infected, and then mutations, lots of mutations, especially adaptive mutations occurring in those chronically infected individuals, and maybe they're being spilled back into the population at large. So that's another possibility. But we think that transmission bottlenecks, these really tight transmission bottlenecks are really important for slowing down population level adaptation of SARS-CoV-2. Okay, and I just want to mention here, this bottleneck work I talked about is currently in press, and there's been some recently, published studies also by Katia Koelle and colleagues, which similarly kind of have found very small transmission bottlenecks using that beta binomial method. Okay, actually, give me 1 second. I actually didn't bring my phone with me right here. I want to see how I'm doing with time.
It's 12:40.
41. Yeah.
12:41. Okay, I can just very briefly mention. I have 10 minutes.
Yeah,
10 minutes. Okay, great. Okay, so I'm going to tell you a little bit about this other work with Youngson Park. It's much shorter, but basically this is more on the phylodynamics work that we've been doing. So as some of you probably know, and this was kind of the one thing that we covered in that perspective piece, is that SARS-CoV-2 sequences have been used to really understand epidemic spread of this virus. So one thing they've been used for is looking at kind of how many viral introductions have occurred into a region using kind of phylogeographic methods. And so we know in many of these cases, like in Northern California, as well as kind of in New York City, and also some analyses kind of in Europe, that there seem to be, and also in Israel, there seems to have been quite a few introductions into a focal region early on in the pandemic, and we know that through the analysis of sequence data. Sequences have also been used to infer the dynamics of spread within a region. So this is generally known as phylodynamics. And so what's interesting here is that you can actually use sequences as depicted here, and the timing of these kind of coalescent events, so when these lineages going backwards in time come together, to estimate things like R0. So this is the basic reproduction number. It quantifies the number of secondary cases a single infected case infects over their period of infection, in a completely susceptible population. And so this is a very important quantity that's been looked at and estimated early on. So this is an example from looking at some sequence data from France. These estimates of R0 over different periods of time. Sequence data can be used for that. And I'll skip this slide. Basically, for those of you who are unfamiliar with coalescent theory, depending on kind of these infer timings of these coalescent events, these timings are informative of the population size, right? And for viral infectious disease, the population size that we're interested in is the number of infected individuals in the population. And so if we think about an SIR type model, so a compartmental kind of ordinary differential equation type model, where you can kind of simulate the number of susceptibles over time, recovereds over time, infecteds over time, under a specific parameterization of that model, like under a specific value of R0, we get a different trajectory, for the number of infected. So if we have a higher R0, maybe we'll have kind of a higher peak and a shorter epidemic outbreak, right? And so, so the population sizes over time are going to be different under different parameterizations, right? And if you have these coalescent timings from the phylogeny, right, they can be used to actually help infer kind of your R0 value. And so we've done this in terms of looking at sequence data from Israel with our collaborator Adi Stern at Tel Aviv University, where we've looked at a few hundred sequences from Israel, and we know that there's multiple introductions into Israel. And using a phylodynamic model that was developed by Eric Volz, we estimated R0 before lockdown occurred, and then the factor by which R0 actually was lowered following lockdown. And what we found is an R0 of about between 2 and 3, depending on how much transmission heterogeneity we assumed, or the extent of super spreading we assumed, and that the quarantine or the lockdown reduced kind of the R0 from about 2 to 3 to it reduced to about a third of its original value. So I'll just mention this very briefly. Is what we've been working on very recently is based on our work looking at the Israeli sequence data. What we found is that there's a huge amount of phylogenetic uncertainty. So we don't actually really know the branching that well. And so if we don't know the branching that well, what we have to do with these phylodynamic methods is actually integrate a lot over that phylogenetic uncertainty, right, which is computationally very intensive. So we thought, well, maybe as a first pass, maybe what we can instead do is use a simple population genetic statistic to actually infer R0, instead of actually using a phylogenetic tree that has a lot of uncertainty in it, or needing to integrate over that uncertainty. And so what we were thinking about and have been working on is using actually the number of segregating sites. So segregating sites, if you look at, let's say we have these 4 sequences over here, we just look at the ones at the sites that are polymorphic, so have more than one amino acid, more than one nucleotide. And so you can see these starred ones are ones that are polymorphic. So the number of segregating sites in a sample is just the number of polymorphic sites. And what we can do is, under certain epidemiological dynamics that we can simulate, we can show that if you just look at these 3 plots, that under different parameterizations, our trajectories of segregating sites in the population is very different. So we quantify segregating sites by saying over these time windows of, let's say, 4 days, let's just look at the sequences that were sampled over that period of that time window, and calculate the number of segregating sites within that sample of viral sequences. And you can see that these trajectories are very different, which indicates that we might actually be able to use these segregating site trajectories to infer things like transmission potential or R0. So for some of you, you might be familiar with kind of particle filtering methods and state space models. So we basically use particle filtering to try to estimate R0 using these segregating site trajectories as our data. And I know I'm racing through this, but I'm happy to chat about it during our discussion afterwards if anyone's interested. And basically what we find is, and we're currently comparing it to actually some of these coalescent-based coalescent-based methods and birth-death process models is that these segregating sites trajectories are actually, hugely informative of these epidemiological parameters that other phylodynamic methods have been trying to estimate. So here, what you can see in this inset is just on the y-axis is the log likelihood. On the x-axis is just the basic reproduction number, or R0, so the parameter of interest. And this is our likelihood curve. And the red line over here is kind of the true value that we use to simulate the mock data set, to test out the method. And the blue is the maximum log likelihood that we kind of, infer here. So you can see that they're very kind of close in value. We've also applied it to sequence data from France. And what we find, we can also parameterize kind of the mutation probabilities and so forth. And what we find is estimates of about 1.5 to 3.0 for the transmission potential or basic reproduction number of SARS-CoV-2 within France. Okay, so I know I raced through that last bit really, really fast. I'm sorry about that. Okay, so just to conclude, pathogen evolution can be characterized at all these distinct scales. We've focused here primarily on looking at evolution that occurs over these transmission events, and also in what I raced through at the population level. And what we're looking at now is actually not only, evolution of the virus and how it tells us about epidemiological spread, but actually thinking about evolution of the virus and kind of its adaptation and using some of these methods to see whether in certain lineages, in certain, if you're looking at Delta, for example, compared to Alpha variant, whether there's differences in terms of the bottleneck sizes and also fitness differences in terms of different R0 values for different variants. So pathogen evolution can be characterized at these distinctive scales and can be very informative of patterns of viral spread. And here I just want to, again, thank Mike Martin and Youngson Park for their contributions to what I talked about and some funding sources as well. And with that, I'll take any questions if I have any time. Sorry for racing through things at the end.
No, this is great. That leaves time for questions for sure. So yeah, thank you. Thanks very much, Katia. There's I see, okay, question from Selena and then Simon.
Hey, Katia, great talk. I guess I had a question about the first part of the study. So is this assuming like we're the 1 to 3 viral particles, you're talking about infectious viral particles, not like based on the particle to PFU ratio, right?
Infectious particles, yeah.
Okay, so it's like more like 1,000 to 3,000 based on like a 1,000 to 1 particle to PFU ratio. Have you ever like incorporated into your models any clustering? I was thinking particularly for norovirus because in enteric viruses, it's thought that like some of the viruses cluster together, and therefore it could be that a non-infectious virus is sort of allowing entry of the infectious virus because this is the one that stuck to the cell, but this one was attached to it. So that's the one that got in and ended up being like the founder virus. Have you thought about incorporating those into any of your norovirus models, for example?
Yeah, that's a really interesting question.
It's complicated.
It is complicated. We have been starting to think about that. I think the simplest model is always to go with, okay, let's think about all these viral particles as being kind of, infectious viral particles as being totally independent. They independently come, from a donor to recipient, and then once they're actually on the, at the recipient, there's no cellular co-infection. There's no, there's nothing like that, and I mean, especially for flu, which we've also applied this method to. It's that's definitely not the case. We know from work by Chris Brook and Anise Lowe and others, that for flu, a lot of kind of complementation has to happen because there's incomplete viral genomes, et cetera, and so, so it's, we are currently working kind of on extensions. The thing is that I mean, part of the reason why this method is kind of used is because it's very straightforward to use. Basically what drops out in terms of the derivation of the equation. So you don't actually need to know kind of in terms of this birth-death model. You don't need to actually even quantify the birth rate of the virus or the death rate of the virus in terms of this birth-death process or the time of sampling. Like all that just drops out, which is really nice.
Yeah.
So that's why it's kind of easy to use.
Yeah.
That said, so part of me is leaning towards, okay, let's take those assumptions and then just, using more of like a thought experiment, see kind of how those results can be off and in what direction they can be off if there is viral aggregation, cellular co-infection and so forth. And I think, part if there's viral aggregation, if it's viral aggregation, but kind of it's in kind of assortative kind of groups, because of where it comes from the donor, then we can be off, and the transmission bottlenecks exercise could actually be much larger. But if these viral aggregates are kind of coming from kind of random samples from the donor, then those estimates should be, using that independence assumption should be adequate, I think. So I think and then if it does come from different parts and there's a sort of kind of mixing, of the viral genotypes in the donor, it's just
it's tricky. Yeah,
it's tricky.
It's complexity.
And I think, it seems like at least, so there's some really nice work by Ben Tenover's lab. It's a Varble et al. Study using bar-coded flu virus. And what they find is that only, a small number of barcodes actually kind of, so you experimentally infect an index animal, right? And then there's a contact animal, either by, contact transmission or aerosol transmission. And so you can see how many barcodes kind of make it across, right? And so from that, they found that in that study, with ferrets and guinea pigs and other small mammals, it's under 10, and so it's not unreasonable, and for seasonal flu, we think it's 1 to 2 based on, the transmission pair data that Macron et al. Looked at. And so that's at least kind of a sanity check. So you can do experimental infections with barcoded virus to kind of, tell you, so I think that's a way around. You just can't do that on humans, right?
Yeah, exactly. Okay, thanks.
Great, go ahead, Simon, and then there's a couple others in the chat.
So the first one is you're using the number of segregating sites. And in humans, we always use like pairwise differences and distributions of allele frequencies. So is that like too noisy in this case to be able to use, or would it be possible to extend these methods to more general statistics?
So we have been, so you were saying the distribution of allele frequencies. So we have actually we've played around with that, and we are working on that. And so I think that's a really great point. In part, it depends also on kind of, so there's some nice work by Chris Illingworth that has looked at estimating transmission. So this for transmission bottleneck sizes, where you kind of assume genetic linkage, for kind of a gene segment. So you're just looking at kind of these different, haplotype frequencies, right? So you can think of that as kind of one locus, many alleles. And so the methods get kind of changed around there a little bit for the segregating sites analysis. We were thinking about looking at the distribution of allele frequencies, and that would be, super, super cool. The problem we kind of hit into, and if you have any thoughts on this or kind of have any papers that can kind of show us around, how to get out of this problem, is more on the calculation of the likelihoods. If we're comparing kind of, so we have this particle filtering approach. So we have each one of these particles, we simulate forward, right? And then at this next time point of observation, we basically kind of sample those simulated populations, and we figure out what for the segregating sites model, we figure out the mean number of segregating sites from these kind of random draws from all these particles. And then we can say, what's the probability of observing the observed number of segregating sites, given the model expectation, which is that mean, right? And so we then give a weight to the particle, and then we do the particle filtering, right? So when we have a distribution of allele frequencies, we didn't quite know how we should assign weights, and we need those weights to actually ultimately get to our likelihood calculations kind of from, so that's what we, that's the sticky part that we kind of, we agree kind of that there's kind of, there's better descriptions, right? Rather than just segregating sites. But we didn't quite know how to get it working quite yet. So we've run into, we still need to think about it, but if you have any ideas or have kind of, yeah, if you have any ideas, I'd love to hear them for sure.
Well, I do. I'll have to do some reading before seeing if they make sense.
Yeah, yeah, that'd be great. Yeah, thanks.
Great, thank you. I think, look, there's a couple of questions in the chat, which I believe are both from students. So, normally I like to prioritize questions from students and tell the faculty to be quiet. And this time you got, in this case, you got 2 faculty questions, but you've got some time with the students now. So I think I will let the questions from Benjamin and Lucas, you can do that now with the students, or maybe give Katia a couple minutes for a break. The students can stay on this link. Is that right?
That's right. Yes, stay on the same link.
Okay, great. Well,
great.
With that, on behalf of QLS and everybody here, thank you, Katia, for an awesome talk. Very interesting. I'm looking forward to chatting with you later as well.
Sounds great.
3:15, I believe.
Yep.
Cool. And I'll send you the link to remind you.
Great. I've got it.
Oh, okay, you got it. All right.
Jesse, we have all the links are all organized. You're the co-host on that meeting as well.
Wonderful. Okay, so I will see you later, Katia. Thank you so much.
Great. Thank you all for coming.
Thanks everyone for being here, and I'll leave you with the students.
Okay, sounds great.
Thank you.
Seminar. Yes, indeed, and leave you in good hands here.